All right, with that, we're going to go ahead and get started. Welcome to another Mass Medic webinar. Today, we are talking about tissue engineered medical products. And today's webinar is hosted by our friends at MPR. My name is Nicole Owens. I'm the Director of Marketing Communications for Mass Medic. If you're not familiar with our organization, we're a medical device trade association that works to bolster the industry primarily in New England through education, events like this, connection, awareness, and advocacy. I do have a few housekeeping notes to go over before I turn it over to our moderator for today. This webinar is being recorded, so anyone who registered for it will receive a copy of the recording after the event today. And we are going to have time for questions. So please feel free to put your questions anytime you have them in the Q&A function at the bottom of the screen, and we will do our best to answer them. All right, without further ado, I am going to turn it over to our moderator today. Maria Pascal is a senior engineer from MPR, and she'll be leading the conversation. Maria.
Thanks so much, Nicole. Appreciate the introduction. So as Nicole said, we're going to be talking about tissue engineered medical products today and the process of taking them from the lab to commercialization so that they can reach patients. I'm joined today by panelists from Epibone or Genesis and MPR Associates. And a quick background on my own company, MPR Associates, is that we are a company of a little over 200 engineers, scientists, and designers. And we're located primarily in the DC and New England areas. We do a variety of work in health and life sciences and in power. Our background is from the nuclear Navy, and we use a first principles engineering approach to take really hard problems and break them down into solvable chunks. Before I have our panelists introduce themselves, I'm going to give a quick background on tissue engineered medical products, since not everyone in the mass medic community is familiar with them. So using the NIH definitions, tissue engineering is the combination of scaffolds, cells, and biologically active molecules into functional tissues. And regenerative medicine is an umbrella term that includes both those tissue engineered materials and individual components and sometimes cells that are used to induce a therapeutic effect in the body. Using our comparison to the construction industry with which we're all familiar, I'll do an analog for a hotel. When you build a hotel, you need a scaffold to hold up the building. And that's like the scaffold that holds the cells in place in a tissue engineered medical product. Then each cell is nestled into a certain location, much like the individual rooms of the hotel. Then you have biologically active molecules like signaling molecules and proteins that are able to move from cell to cell and impact the functionality of the different cells. That's like the room service bringing what you need to your hotel room. So with that brief introduction to tissue engineered medical products, or to the terms tissue engineering and regenerative medicine, I'm going to speak about both using the umbrella term tissue engineered medical products. Thank you for sitting through that brief introduction. And now we're going to have you meet our panelists. First, Ik, could you introduce yourself?
Hi, my name is Ik Umratana. Sorry, you can pause it for now, Maria.
Oh, sure.
I'm a Chief Scientific Officer and co-founder of Epibone Inc. We are a tissue engineering company growing skeletal tissues for repairing body parts. And our main component is a stem cell-based tissue. So I will give an overview of our technology. So our company use either CT scan or imaging informations to create scaffold and bioreactor from these images, so that we can design the specific tissues that the patients need. Scaffold is being made, cells is being grown and placed inside the bioreactors. The left is the bone bioreactor, the right is our osteochondral tissue that we are growing. Stem cells in these bioreactors can differentiate depositing tissue matrix of specific types, and over certain amount of time, these tissues become more mature and ready to be implanted into the body. To date, we have 3 product lines: a tissue engineered bone, tissue engineered osteochondral grafts, and a cartilage, an injectable version of the cartilage, and we are aiming to push this toward repairing many joints in our bodies, many bone pieces, and also giving patients a better life. We are clinical stage. Our bone product is in clinical trial. We completed the recruitment and implantation. Our following product is in the process of IND submission, and we are looking forward. Hopefully, we'll get those products in the patient soon.
Thank you so much, Ik, and thank you for speaking to how you start with a scaffold and with cells and end up with a product that's ready to be implanted. All right, Ryan, could you introduce yourself next?
Sure. Ryan Downs, I'm with MPR Associates. I'm Vice President of Health and Life Science Practice Area within MPR. My background's mechanical engineering and automation. I'm also particularly interested in technology, strategy, business modeling, and automation. A little bit about MPR. We have 3 core product or service areas where we help clients achieve their objectives. At a high level, they're project execution and risk management, which is helping plan out, de-risk, and execute very complex things. We have a core capability in product and system design and development, helping implement those plans, whether that's developing a medical device, designing and developing a process, organizations, a facility. And then asset management and reliability is more around keeping existing things going. So if you have operating equipment, more sustaining engineering, things like that. So just in a nutshell, that's a little bit about what MPR does. And we try to customize what we do to kind of fit the need. We see temp engineering as a growing area in the future, and we're happy to participate in this kind of thing. Thanks.
Thank you very much, Ryan. And Paul, could you close out our introductions?
Sure. Thanks, Maria. I'm Paul Kosnick. I earned a PhD from the University of Michigan in 2000 in biomedical engineering. Little trivia, I was actually the first person to graduate from their concentration of tissue engineering, which was a new concentration they had within the PhD program there. From 2000 to about 2005, I held a number of academic appointments, including 3 years as a research scientist for Harvard and MIT in their Human Sciences and Technology program, and helping several universities develop biomedical engineering curricula, including UNC in Chapel Hill and University of Hawaii. But my life's work has been developing devices for cell therapy, and a big part of that was developing the device that you're watching now, which is known as the isolator. This is a device that isolates stem cells from adipose tissue at the point of care in about 1 hour. I was the principal investigator, and I've been awarded over $32 million in grant funding to develop the isolator, carry out preclinical studies, develop a clear regulatory path, conduct several first-in-man studies, and bring the isolator into cGMP manufacturing. So I've been involved in the process from napkin drawing all the way to the bedside. Obviously, I'm one of the co-inventors of this technology, along with my colleagues at Tissue Genesis and also MPR Associates. And Tissue Genesis was acquired about 2 years ago by Orgenesis, which trades on the NASDAQ under the ticker ORGS. I currently manage Tissue Genesis International for Orgenesis, which is a division of Orgenesis. What you're watching is a video of the isolator. It's functionally closed and fully automated. It's an isolation device that is OR compatible, cGMP compliant. It isolates tens of millions of viable nucleated cells up to about 100 million right at the point of care. And those cells can be administered back to the patient in about an hour for any number of different indications. It requires no special training to operate. It's best in class for efficiency of isolating stem cells from fat. And it's been approved by the FDA for 10 clinical trials in the US, both as a device and a biologic. So we have walked both those paths. We currently ship the device and consumables, which include a plastic disposable kit and a reagent, to clinical trial sites in the US and limited commercial markets around the rest of the world. So it's been principally my life's work for the last 20 years. And that's, I guess, what I'm here to sort of contribute is that experience.
Thank you very much, Paul. So now you have context that Epibone and IC are in their clinical trials, and they're creating a product that takes cells from the patient and then implants a full product back into the patient. We've heard that Paul is on the other end, where they've had a product on the market for almost 15 years that works at the patient's bedside. And then MPR has worked with companies, including Tissue Genesis, who are already at their end stage, but has also worked with other companies who are at different stages of the process. So we're going to give you the full range during our panel today. And with that, we're going to jump into our first question. So some of the commonly cited roadblocks for the commercialization of tissue engineered medical products that I hear with regularity include chain of custody, availability of media or cells at a large scale and an affordable price, availability of a skilled workforce, or the need for large amounts of skilled manual labor, contamination, real-time sensing during production and transport, and the knowledge of in-process quality, critical quality attributes to enable monitoring of product prognosis throughout the production process. So for each of you, as you are companies which have commercialized or are in the process of commercializing TEMPs, can you talk a little bit about your own challenges from the technical standpoint, including one of your acute challenges? And it would be great if you could start.
Oh, all right. Let's see. So I think I can easily speak of from when we spun out from the university. We spun out from Columbia University in New York, and we are still based in New York. So it's been the past 9 years now that we have been running this company. And during these times, we are transitioning from the very academic research work into the R&D lab, so to speak, preclinical lab that we run at Epibone, and now transitioning into a CGMP manufacturing, which we are having a small facility in-house to perform these manufacturing of human clinical product. So the challenge from transitioning these step process, one of the major challenge is from learning and understanding the supply chain and logistic of this. From early on, we always grow small few pieces of grafts, and as we develop our product and toward clinical trial, especially clinical trials, we have to plan out how are we going to manage these whole supply chain, right? Like every single materials we have to have in the facilities ready to manufacture. So along that process, there is a lot of strategic thinking that we need to make decision on. Early on, we can buy a few bottle of media, a dozen bottle of media. And when we start transitioning to a media that is more specific to our process, we need to start working on thinking of contracting and working with the manufacturer, these suppliers. And there are a wide range of suppliers in the US, which is a luxury, but also a decision for us to make, right? There are smaller one where they can fine tune the product, but they also have less experience on getting their media into the drug master file with FDA and our confidence in making sure that those media works. And then they're also a bigger company where they always supply the media in multiple liters. There were conversations where do we buy 100 liters of media or do we buy 5 to 10 liters of media, right? And the media is the most expensive materials for basically TEMP development. So these we have to work through every single material list from the media, from flasks. During a couple of years ago, there's a very limited supply of tissue culture flasks, and we have to be one of the hoarder. So we have to think along what happened if we need this within a year. There's expiration date on all of these media and flasks, and try to set it up. We want to be frugal, but we cannot be frugal that result in us not being able to produce. And there's looking at the supply chain alone, it is something that a new product developer, I would say, really need to consider early on and really look into, discuss with the supplier, how can they support us? How long can they support us? Can you hold certain amounts of media to support us throughout the whole next couple of years? And this is one of a challenge that we have to always think about, and making sure that we are ready to manufacture these products for patients.
Thank you for speaking to that. So to summarize, the challenge that it just spoke to was the challenge of scaling the inputs as the production increases from those really small scales to scales that you need as you move towards commercialization and clinical trials. So thank you very much. And Ryan, how about you? What's one of the biggest challenges you've faced? Oh, I think you're muted.
So we see a lot of, we come into the a lot of development efforts after the sort of core science is formed, the business model is kind of understood, and then there's this large gap to how do you get there? How do you get there at the scales that make the business make sense, that the products are profitable, that the cost of goods are where you need them. And in TEMP, it's particularly challenging because you're often talking about allogeneic samples where you can't mass produce. Everything's a different flavor of a one-off. And there's in our experience, there's two ways to scale. You're either going to scale in a very manual perspective. You understand it takes 2 people and 2 days to set up a process, and it takes 3 months to work out a solution. And how do you compartmentalize that and replicate? And then you're going to need hundreds of people of certain quality. So labor force, skilled labor force is a key in that kind of scenario. Or you go the process of automation, where you really try to focus on what are the important characteristics of the process, what are the key process parameters, how do you distill those down, how do you remove the human from and the skill and the art of the human from the process, and make a process, or in the case of something like tissue genesis, a closed form product that replicates all those steps and does it with sufficient reliability and speed, where you don't need the workforce, you can reduce the time, and you can maintain or increase the quality of the product at the end of the day. So, and in certain cases, that's the way that you have to go. And in some cases, clients will go do both. They will scale up to a point, up to proof of concept, up to proof of market, up to where they start to hit the need and the growth curve, and then shifting to an automated solution starts to make sense. So in a lot of cases, automation and scaling is all of the above.
Thank you so much, Ryan, for speaking to how the effort needs to scale, and that effort can come from people power, but can also come from equipment power. That's a great insight. Paul, can I turn it over to you?
Yeah, thanks. So our product is not a traditional tissue engineered medical product, but we're part of regenerative medicine, cell therapy at the point of care. So we do face some of the same challenges, but our manufacturing process actually happens bedside, right? So the device operates in an austere environment or a semi-austere environment. We don't have a biological safety cabinet or a hood there. We may have people who have limited training with respect to aseptic technique and some of the things that would be required for traditional manufacturing tissue engineered products. So we face a number of different challenges. One is because we're in that austere environment, we typically have to go automated. It's not an option to have a fume hood, a hood in the OR or somebody with a pipette. So automation is critical. It's one of the key hurdles, but also one of the key enabling technologies and enabling to put our platform there, right? So that was very important for us. Another thing that's important for us is that we don't fit the traditional paradigm for either a biologic or a device. And the regulators struggle with that. And maybe even more importantly, long-term, third-party payers struggle with that. So in a traditional device, you have something that's either used externally or implanted in a patient. Regulators know how to regulate that. They also know how to, third-party payers know how to reimburse for that. They know what the longevity of it is, what the benefit to the patient is. With a biologic, you have similarly something that's, implanted. And again, it's fairly well understood, similar to a drug, but living. So it fits kind of a drug paradigm in a lot of ways with just a little bit of adjustment. When you're delivering the patient's own cells back to them at the bedside in a single session, a single procedure, a lot of those rules go out the door, right? One of them is quality assurance or quality checks that you can do for your product. If you manufacture the product over a month-long process in a lab, you have plenty of time to measure a lot of different metrics and tell the FDA a lot about both the performance and the safety profile of what you're providing back to the patient. When you are manufacturing the cell product at the bedside in a matter of an hour or two, you don't have that luxury, right? So you have to come up with quick metrics that can be meaningful, but can be done in a way that the cells, while they're still viable and while the patient is in the same procedure, to be delivered back to the patient. And then similarly, again, reimbursement. At the end of the day, we're not going to be able to get our products out to the greatest number of patients who need them if we can't navigate how we get reimbursed, right? And the third-party payers just don't really understand, again, not exactly a biologic, not exactly a device. The FDA regulates us as a device or a biologic, depending essentially on how we prepare an application for them and what claims we make. So those are the sort of critical things that we needed to figure out. In some cases, we were the first to figure out and develop a path with the FDA. And so they're both hurdles, but enablers, right? Once you do conquer them, they're what allow you to get out there, get into the marketplace and start helping sick people, which is, I think, why we're all in this industry.
Thank you very much, Paul. I appreciate the distinction between a bedside therapy and a therapy and a product that has a longer time scale in order to prepare. And I think that's a great transition from the technical issues to approaching the topic from the regulatory perspective. As you mentioned, you were one of the first companies to do this. And can you speak a little bit to how the regulations have evolved over the last decade?
Yeah, sure. So we got our isolator device approved for a clinical trial in the US, first in man study 2009. We were the first bedside device to get an investigational device exemption from the FDA. We've since grown that to 10 studies that have been approved, both as devices and also as investigational new drugs as a biologic, again, depending on how we kind of administer the cells when we're done. One of the interesting things that's happened over the last 15 years is the FDA has wised up to this. We've seen a lot more guidance. We've seen a lot more standards published. And Europe has actually done a complete 180 in terms of how they regulate these sorts of products. With all of that new understanding comes new regulation, right? And so there's now more work for compliance. Generally, I think it's been great, frankly, to have better understanding, although it creates more work. Again, generally, the work is both necessary and welcomed. It's providing safer products. It's providing more reliable products. It's better defining what we deliver. And I also think, again, hand in hand with regulations is reimbursement. If the regulatory environment is not well defined, third-party payers are reticent to pay for products, right? And so, although we've seen a great, essentially kind of an explosion in terms of, regulatory documentation and burden with these kinds of products, generally, I think it's a good thing. I think we've gotten definition, we've gotten the FDA to understand how these products need to be regulated, what makes sense. We've moved the industry forward, and we're getting closer to being able to provide many different treatments at the bedside and to have them delivered in a safe and efficacious manner, and to be able to get reimbursed for them, again, so that we can reach as many patients as possible.
Thank you very much, Paul. And I see a question from Evan in the chat that's related to this, asking about how TEMPs are related to human cell tissue and cellular or tissue-based products. And panel, feel free to chime in, but my understanding is that this is different nomenclature talking about effectively the same thing. So thanks for the question there. And the industry, being as young as it is still settling on exactly what language to use. So thanks for that question. And now that we've heard a little bit about—
At the risk of disagreeing with you—
Sure, go for it.
I actually think HCTs are regulated under a completely separate division of the FDA.
Okay.
So this is more toward tissue transplant type regulations. So if I think I understand this correctly, these are products that are— So when you hear somebody say a product is FDA approved, the technical jargon is it's been cleared, and what we call a pre-marketing application has been applied for, and the FDA has approved it. On the basis of the package you put together, they think that the risk-benefit ratio is reasonable. With tissues, this is more like tissue banking. And those products are not pre-approved so much as the FDA publishes regulations, and anybody can basically set themselves up to operate in that space, and they follow the regulations. Those are marketed as HCT products, but not pre-approved. So they technically can't say they're FDA approved. So for example, there is some overlap here, and there's some gray areas. And in fact, there's some areas where the FDA has made a distinction, which is somewhat not consistent with their own rules, right? So I'll give an example. It makes sense, like, let's just say organ donation, and you collect an organ, you have a tissue bank, and then it goes somewhere, it gets transplanted into somebody, right? This all makes sense. We understand that from the sense of both auto transplant and transplant from one person to another, right? But there's some areas where the FDA will allow some of those organs to be manipulated a bit. In some cases, they can even be like enzymatically digested, shifted around a little bit, sent back, and the FDA still regulates that as an HTP. So for example, I happen to know islets are regulated that way. So you can take a pancreas You can isolate the islets, deliver them, administer in a different facility, administer them back to the patient. The FDA considers that a tissue transplant essentially and regulates it here. Whereas if you were, manufacturing islets, that would be a biologic, and that obviously would be completely different.
Thank you very much, Paul. And Ick, it looks like you might want to chime in on it.
Yes, I actually have a very direct experience in this because we and since we are in the regulatory field, so we are almost like an extreme end of the process from what Paul has worked on. We take the cells, we manipulate it heavily. We would say, to the best of our knowledge, it's a regulated FDA clearance tissue engineered products that differentiate stem cells. So we differentiate stem cells and reimplant into human. This is not minimally manipulated. So HCTP is when you take out and put it back in a similar environment, or you very minimally manipulate it, right? So for us, for tissue engineer TEMP, if you manipulate it, expand the cells, we are not regulated under HCTP, and we'll have to go through IND, and CBER is very clear with that. So that's the process that we learn. It is a more challenge process, and talk about challenge from the FDA that this is the regulation that they set for good things. So they want to make sure that any manipulations doesn't alter the cells, doesn't alter the biological too strongly, or alter it to the level where it can be adversely impact once it planted. And what FDAs care about is the safety. And our experience working with the FDA is we feel that FDA is very collaborative. They know a lot about this field now. Maybe not back in 2009 when Paul started. We started in 2014. We get clearance in 2019. They know what this product is about. They know they have specific sets of data they want to look for, and our job is to work to get those data and present those data so they feel comfortable that the product can be safe and effective in treating, and the risk is much lower than the benefit that it provides. So, and when it comes to, so that's the first part of getting the clearance through the IND process from the FDA. The second part, which is nowadays becoming a challenge, a big challenge for us, is the indication of this product, because we are not HCTP, and so we grow bone, but it doesn't mean that we grow bone and it can be placed anywhere in the body and right next to other piece of bone. They look at the indication where certain indications are more similar, certain indication is very different. I'm going to give examples. We can now implant our bone into the mandible regions, but if we want to go into the cranium, if we want to grow an anatomical cranium place, we definitely need to provide additional safety data, because cranium is right next to dura and so on. So that's the way FDA is thinking about this tissue product, because it's not, yes, we call it a tissue engineered bone, but it's not exactly bone. It's a cell manipulated tissue engineered bone, right? So they still think there is some safety concern that they put in place. Hopefully one day they will feel more comfortable that bone can be placed in bone, cartilage can be placed in cartilage, tendon can be placed in ankle and knee and wrist. But today, that's how they are looking at this regulation, and they're gonna look at the data that we can supply to them and giving us green light to expand the indication. I think there's a lot of work from our side, from the developers, any developers, to prove that it can be expand beyond just certain indication, but that is also on our side to keep working on.
Yeah, and Maria, I would refer Evan to 21 CFR Part 1271, Section 361. That's the regs for the HCTPs.
Yeah.
There's
Evan,
a
I
pretty
hope
good
you wrote that
definition
down.
right up front.
Great. Thank you both for providing insight into your own regulatory processes and into the difference, even though they're all cells or tissues, into the difference it makes to the regulator when they're minimally manipulated versus more heavily manipulated, and the heavily manipulated falling more under the nomenclature of TEMP. So definitely great insight there. And Ryan, can you speak some to how in the various stages of the commercialization process, MPR can help ease regulatory restrictions for some of the companies that are going through this?
Yeah, I would say probably the most important thing to do is to have a development roadmap. So what are you trying to achieve? Over what timeframes are you trying to achieve it? When are you going to engage the regulator? You're engaging the regulator on a manual process, or you're engaging the regulator on an automated process, all of those things, in addition to everything we were just talking about what's the underlying biology and how are we manipulating things. I'll say the technology, techniques, and processes that go into it outside of the body, outside of the biology, are extremely important too. So the roadmap is one. I think understanding how you can minimize contamination in this kind of environment is very different, because typical other medical devices and processes, just a good old-fashioned sterilization, does the body good. But here you have a bunch of living cells that you're trying to keep, and also keep the bad stuff out. So really trying to understand contamination sources as you bring these cells outside and you manipulate them. That is key, because you don't have the benefit of just sort of mass sterilization. And when you design equipment and processes, thinking about what, if it's an automated system, what are your disposables, kits, and what are your durable infrastructure? If you saw in Paul's video, they had that nice little kit that went into the machine that had the centrifuge, and that was a very thought out contamination prevention concept, right? So that you can keep the core, the base device, out of the biological zone, and everything in that consumable inside the biologics to minimize contamination. So I think doing those sorts of things and having clear cases, documentation, and test experience is one way to probably the best things you can do to help the regulatory process go smoothly.
Thank you very much, Ryan, and appreciate the kind of foreshadowing that I'm going to use to transition us with having our roadmap as you're going through your production, and latching onto that desire to have a roadmap at the beginning to determine what you're going to do at each step of the way. For Ik and for Paul, when knowing all of the challenges up front about the technical and the regulatory hurdles, commercializing could seem daunting for some of the people on this call. How did your companies decide when to scale up your production for commercialization, and when did your means of production have to change to meet your new scale? Ik, do you want to start?
Sure, I can start. We think it is very important to short answer. So we start quite early. As you've seen in the video, we design the bioreactor, and what Ryan was talking about is how do you design something that is very simple and kept everything sterile, right? And that's when it's simple, it allows scalability. So the bioreactor that we design, and it's a common concept now, is there is a disposable portion where it's cheap, it's easy to build, it's easy to put together. Once it's done, you throw away. You don't have to do a cleaning or cleaning validation of those. then the whole system itself is outside, that is never touched the product. So we do think about this very early on during the preclinical, because we want to make sure that the concept of tissue culture doesn't change a lot, because when you change a lot, that quality attributes may change later. So we spend a lot of effort in designing the bioreactor system, how the cells are receiving nutrients, how do we know they are receiving nutrients, using computational simulation, using measurement, sensor measurement, force, flow, pressure, and also the biological measurement. So we do take that input early on, because we know that as we develop the product through preclinical, early phase clinical, and by the late phase clinical, the process can change. But as the process change, we still have to prove that the product is still the product, which means that the cells are still acting the same. The protein that cells are making are still the same. The quality, the mechanical properties of the product, whatever your quality attribute is, that has to, the data that we collect now will support the scalability in the future. So we focus on the data a lot. The design will change, and we know that the design of the system will change. We're gonna be scaling. Right now we grow 1 at a time, then we grow 2, 4 at a time during clinical trials. By phase 3, we probably need to go 10, 20 in a month. The quality attribute dictate the scalability of the product, and putting that in place, measuring the quality, measuring the quality early, and thinking to what's the easiest way to make the product, to grow these TEMPs, is we start from the preclinical.
Thank you. Thanks for speaking to how Epibone is planning with the end in mind, knowing that just to get out of clinical, you're going to have to be doing 30 pieces of product in a month. And that's a 10x scale from where you are now. That's great to understand.
Yep.
And Paul, so TGI closed and automated your cell isolation system over 10 years ago. And that was well before it was popular, and organizations like the Advanced Regenerative Manufacturing Institute, which Epibone has collaborated with, some existed. And are you seeing that some of the challenges have now been mitigated for the generation of TEMPs coming through the pipeline?
Yeah, I think, I touched on it earlier. So at the risk of repeating some of that, when you're first, the regulators, third-party payers don't know what to do with you. You might think that's great. That means maybe you get to make the rules, but typically, because these institutions are very risk averse, it just means you have to sit around and wait for them to catch up, right? So it's not quite the advantage that you may think to sort of be first out there. You, as my old boss used to say, you take all the arrows for everyone that comes after, right? And so I do think the industry is in a much better place now. And Ick has touched on it too. The agency understands now risk benefit, and the risk benefit specifically of these kinds of products, and how to manage that and how to balance it. So I do think that the industry is in a much better place. To touch on this manufacturing issue, we are going in the sort of other direction, right? Where we're going right at the bedside, at the point of care. We're manufacturing one product for one patient in that regard, with respect to the cellular product. But we still face a lot of the exact same issues with respect to manufacturing our device, right? So we still had to develop, we have the hardware, the actual device that sits on the countertop. We have that disposable kit similar to what Ick has in his process. And we have reagents, again, similar to what Ick has in his process. Our process, those things go right into the OR. So they definitely have to be made to those rigorous standards, and we definitely have to be able to scale the manufacturing of them. So we share those we share those issues and challenges, even though our actual cell product is manufactured at the bedside.
Thank you. And at this point, it's a good opportunity to start throwing your questions in the chat if you have any. And I have other questions that I can ask, but if you have anything in particular you would like to hear about, now would be a good time to put your questions forth. So thank you for that. And while people are throwing questions in, Paul, I want to ask also, as a company that has both tools, reagents, and creates your own cell product, my understanding is the Orgenesis parent company is more on the tool company side. And how do you encourage companies to use your tools for their clinical trials? Because as Ick was mentioning, it is beneficial to move towards your final manufacturing early in your process if it's readily available.
Sure. Yeah. So we do a number of things both locally at TGI and in what I like to call the mothership. We actually have several divisions. So we're part of Orgenesis, which is a part of Orgenesis. And in Orgenesis houses all of the therapies services. So cell and gene manufacturing services. And so at that level, the company specializes in helping somebody go through that process that Ick was just describing. And that is, a smaller company that has a great technology, inventors are on board. They've, developed something that really has a lot of promise. They have not taken those arrows of bringing a product through regulations, through scale up, and they would like somebody with that expertise to just come in. And so we can take a process that is a laboratory process, and we can put quality assurance around it. We can put ISO standards around it, and we can turn it into a process that's actually CGMP compliant manufacturing process, initially for 1Z, 2Z kind of volume, and then as appropriate for scaling up the volume. Tissue Genesis role in that specifically is we're the home of all things medical device and what I like to call medical device related within Orgenesis. And when I say medical device related, there's this interesting space that a lot of the instrumentation that you use when you're developing a cell therapy or a tissue engineered product in the lab, it's highly regulated, even though it's not technically a medical device. It needs to meet biocompatibility standards, and it needs to meet, pyrogenicity standards and a number of standards that have been developed specifically for medical devices, even though it's technically not a medical device. And so our experience has given us a lot of understanding of what needs to be done, both to be compliant in that space and then also to automate in that space. And I think automation is key ultimately to driving costs out of these and bringing these treatments to the masses. So if there's been a theme to, my commentary here, it's that, our goal is to get these cell and gene therapies and tissue engineered products to as many people as possible. Currently, they're very expensive, and it limits the distribution of these treatments to not everybody who needs them gets them, frankly. And so our main goal is driving cost out. We do that mostly by automating and making these processes streamlined and then getting them closer to the end user. And those are the ways that we drive costs out so that we can reach more people who need these treatments.
Thank you very much, Paul. And I'm not seeing any questions in the chat yet, so I'm going to go ahead and ask for each of you on the call, what advice would you give to other temp producers who are approaching commercialization? Is there anything you would change for yourself or your company if you could go back? Ick, do you want to start us off? I think you're muted.
Oh, sorry, very noob there. There are two I would say two areas. I think I can share the technical part of the products, and then the experience of transitioning from academic to clinical stage company, running the clinical stage company now. From technical side, I would say is if going back right after academic, we are excited about the product. We like, this is a great product. It has proven, we are putting it in animals. But back in the day, we do not think enough about the long term. And one of the thing is the logistics. Working with medicine is not trivial, it's not simple. Hospital has a way of how they work. Surgical setup is very unique, and it's very on point in time. Tissue engineering medical product is a challenge in itself in manufacturing. It takes a long time to do. There's sales control the schedule. You don't sometimes. So where in that process can you be flexible? If you think at certain area can be flexible, obtain the data, provide that data that you can have flexibility. For example, shelf life, simple thing like shelf life. You want something that's viable, you want something that's living, greatest quality, obviously you take it out from bioreactor, implant it the next day, right? But from the reality, that's not a simple task, right? Because you are limited by a lot of logistics, shipping, doctors, surgical. Not every doctors perform surgery every day. That's a knowledge that we learn too. They do specific day of the week, right? So we have to schedule everything from the start, getting the cells from the patients all the way to that whole schedule has to be made. If you don't have flexibility along the way, you're gonna miss it, right? And shipping, if you can have shelf life of 3 days is better than 1 day. 1 week is better than 3 days. 1 month is better than 1 week. So if you can prove that product can have longer shelf lives and still viable, have the quality, that is golden. So I think that's a one thing that if we were go back, we would look at these much earlier on, and it's gonna make your life much easier. So to speak, and that's a technical part of it. From the experience part of it, I think there's so many things around us, right? We talk about supply chain, we talk about automation, we talk about all the companies that play a role, an expert that play a role in these technology. We know what we know best is processing the stem cells, growing tissue, making tissues. We can't do everything else. So we have to work and always keep in mind, know what you don't know. There's a lot of things that we do not know, but oftentimes we don't know that we don't know. That's a complex sentence, but it is true. So once you know what you don't know, you can find the right people like MPR, the right people like all the consultants, clinical consultant, regulatory consultant, manufacturing consultants. Those will be the one that will help because they are an experts in the area, and that will allow us and allow you to focus on what you do best is to do science and develop the product.
Thank you for sharing both of those challenges, Dick. Ryan, do you want to go next?
Sure. I got 3 things, but I'll be super quick with the 3. One is around critical process parameters. You know, what we see a lot of is people know, you know, this process works when I, you know, do this step at a certain temperature or a certain pressure or a certain rate. But one of the things that they don't know is what latitude do they have around that value. You know, they know that value works and they write it in their SOP and that's what they do. When you're going to scale, one of the things that you should really explore is like what range is acceptable around that number that gives you adequate results so that you have a lot more latitude and flexibility as you scale, because by scaling, you're going to introduce a lot of other constraints that you might need to compromise on some of your CPPs. So exploring the range around a CPP is important. 2, you got to understand your business case. I think Paul said it very well, where, you know, he's doing N of 1 every product is an N of one. So in order to do that efficiently, you have to understand I'm bringing the equipment to the patient, and any other way is just not going to fly. So having a really good understanding of what your business model is, and whether it's you can store bone on the shelf for a week, a month, a year, or I got to take my equipment to the operating room. Those are two vastly different, business cases in the same industry. And lastly, I'll leave with tackle the hardest thing first. Too often, people focus on the things that are fun, easy, quick, gives yourself a false sense of accomplishment. And then you spend a lot of time and money working your way, you're painting yourself into the corner when you finally get around to tackling the thing you've been avoiding all along. So I would say in any process, start with the thing you're least confident about and get that locked down and solved, because it gives you the most latitude in terms of design space because you haven't locked in anything else. And then all the other easier things you could pack around your solution for the hard thing. So those are my three wise words of wisdom.
Thank you so much, Ryan. And Paul, do you want to close us out? I'll say you have about 90 seconds before we have to give it back to Nicole.
Sure, yeah. Those were both great responses. I'll play off both of them a little bit and maybe surprise viewers, but I think the most important thing is management. And it's not the technology, it's not the insights, it's not the flashy invention. It's management, both at the very top of an organization, because all organizations I've been involved with are always top down. And it's also management at the project level, right? The world is full of amazing ideas that never amounted to anything because they weren't well executed. And so to play off the commentary from both the other panelists here, I think focusing, if you have a great idea, you have a great invention, you have a great technology, I think focusing on management, and to Ick's point, if that's not your strength, bring someone in for whom it is a strength. And having a clear vision from the top of the organization to the bottom, and then having people who really know how to execute will make you successful, even with a mediocre idea. So if you have a great one, you're really setting yourself up for success.
Thank you all very much. And thank you to everyone who joined us for this conversation about how to take products from the lab to the patients who need them. We hope you learned a few tricks along the way and feel welcome to reach out to us if you have any further questions. Nicole, I'll hand it back to you.
All right. Thank you so much. What a great and really interesting conversation. Just listening is a fly on the wall. It's really exciting what you guys are doing. So thank you for sharing that today. I hope everyone who joined the call today, joined the webinar today, enjoyed it as well. You are going to have access to this program on demand. And please go to massmedic.com/events and check out all the other great events we have coming up in the future. Thank you again to our sponsor for today's webinar, MPR. We cannot put on great educational content for our members without partners like you. So thank you. Have a great day.
Thank you.
Thanks. Bye, y'all.